it has a tendency to do that, but I've never seen Isodella like that with that huge gull.
From this angle, it looks like the sponge grew out of the coral stalk.
Yeah, it's really confusing.
Yeah.
Yeah. Do a really quick partial on this guy. That rocks, yeah. Kind of looks like it though still, right?
Yeah, must be just behind it. Stalk.
Interesting.
Yeah.
Yeah. All right, pull wide, please. I'll get out ahead a bit.
Ship has completed its move. Argus has some little bit ways to go.
Oh, here's another
30
fish.
meters.
Are these tube anemones or urchins that we're seeing kind of on the sediment there a few times now?
I saw a couple of the small red ones. They're not tube anemones. They're just regular anemones. Tube anemones usually signifies a group called the cereanthids, and they have very distinct tentacle morphologies. They have 2 rings of tentacles, one closer to the mouth and one on the outside of the oral disc. So I don't think they were cereanthids, but they might have just been anemones that live in the sediment or on the sediment surface.
So interesting view here in Argus, as well as Herc. You can see that we're kind of coming up a sheet flow that's relatively sedimented over, but you can see in Argus's view that we're kind of in a little sediment chute almost, and that the rocks to the right and left of us seem actually a bit clearer.
Take, please.
I'm looking at this bamboo coral thing now. I mean, it could be a live anemone, but I feel like there's some, it's the wrong texture.
For those who are wondering what Steve is talking about, he's looking at some of the stills from that cool coral gull that we just saw.
Yeah.
Little fish there on the right.
It very well could be an anemone that's
fish
just
or shrimp?
closed in.
Like a shrimp.
Yeah.
Kama, for folks wondering what resources there might be for new students learning Olelo Hawaii, do you have any suggestions?
Oh.
Yes, we do. So actually there are a few ways that you guys, if we're like beginning learners and stuff, and so one of the things that I can think of off the top of my head is you go to YouTube, there are a series of materials that were published within like from anywhere from like the early 90s up until like actually more recent times during COVID. One of those, sorry, so I'm gonna actually pull them up.
Go do a really quick partial on this guy. Nice.
I should probably preface this with saying that there's generally no second to being able to actually join in a class or like with a native speaker and stuff like that. But I realize that might not be totally possible.
I found some of the little apps you can download on your phone for language learning.
Oh,
somewhat helpful.
Yeah.
For learning kind of simple words.
Yeah. Yes. We've seen some of the crew using Duolingo. That was a good app. I'm looking at YouTube right now, and there's a series called Kulaivi, Kulaivi, Hawaiian, and that was actually put out by the Kamehameha schools in the 90s. More recently, we have, shucks, I'm trying to think, but you can also, there's also like the Hawaiian dictionary online too, and that's available at either ulukau, ulukau.org/vehevehe, WEHE, or you can go to the University of Hawaii online dictionary at hilo.hawaii.edu/vehe, WEHE. And if I think of anything else, I'll drop a comment.
Thanks, Kalamayo. Renee, did you say that the ship move is done and Argus should be finishing up soon?
I just called in another one, but
okay.
I can stop that if you like.
Yeah, if you could ask
Sure.
them to pause. I'd like to spend a little bit of time just poking around here.
Sure thing.
I don't think that we've collected this kind of material yet. Kind of want to see if there's anything. So Jess, yeah, if we...
The rocks up to the right, is that the
yeah,
goal?
probably we're going to have more luck over like in here of finding something to pick up. I just want to see if it looks representative of the material above it.
Roger that. Is that a white crab over there too?
What's the white crab at the
yeah,
base of the...
that is. Yeah, we can image that first if you want.
Yeah, sure thing.
It's a white crab. We'll see.
We're not sure what.
There's also another type of squat lobster called Munidopsis in this area too, and this one looks more like Munidopsis, but we'll see. Yeah, it's very subtle differences. The thing I'm looking for is that this one lithodid has a very long rostrum spine.
Go ahead and push on in there a bit, Jeff.
Doesn't want to get picture.
Really?
Yeah.
Oh, you know what?
This does look a lot like...
I think that might be it.
Yeah.
Let me see.
3 walking legs.
Of course.
Yeah,
right.
It's a squat lobster.
He'll come around. He'll come around. Maybe.
Come on.
Or he won't.
Oh, yeah. Wow, that's cool.
Camera shy.
All right, you want to come a little wide there and I'll try to creep.
I'm about 90% convinced it's not the one we're looking for, but we can take a look. Good to be absolutely certain about those things.
Yeah.
It's really beautiful texture on these rocks behind the behind all the animals.
Kind of a rippled texture
for
almost.
the rock lovers in the audience.
All right, let's try number 2. Go ahead, I'm gonna land it, but
yeah, this is a type of squat lobster. It's not
what we want.
Not the crab we were looking for.
Reg.
But still a good observation. Yeah, so this is probably in the genus Munidopsis.
Roger that. Okay.
And then Lindsay, just I found that other resource. The series is called Kaleo OEV, KA space LEO space OIWI. That's going to be found on the OEV TV website, and then you can watch a series of videos. The video series themselves are based on a system for like learning Hawaiian language called Nakai Evalu. That was published out of like by, what is it called? Halekua Mo'o is the name of the press and stuff like that. But so you have these series of videos that have interactive, there's basically a video of the lessons book by book. And then you'll also be able to, at the bottom pane of the videos, download worksheets and stuff like that that might be able to help you guys out.
So Beth, any of this looking in front of us like a good candidate for a sample?
Can you try this piece here?
Oh, yeah. Do you want a quick zoom on it before we do?
Yes, please.
All right, Jeff.
Actually, I'm changing my mind.
It's
Can
pretty
you back
big.
out?
Yeah.
But it's not that, it's just the texture is not quite what I want. Can we look at this one over here?
All right, go ahead, Jeff, and push on in. Take one bump down.
That one looks like it might be attached, actually, so that's probably not a good candidate.
Never know until you touch it.
That's true.
Are you looking for a smoother, more bulbousy, or?
Yeah, I want more bumpy.
More bumpy.
Yeah, can we just try it, Jess, and see if it'll come off?
Oh yeah, we can try anything.
Should be pretty stable.
Roger that, thanks, Ernie.
For our scientists ashore interested in rocks, welcome your advice. You see a better candidate that you would like?
We're looking for botryoidal textures, is that correct? Oh, nice word. Go ahead and push on in there a bit, Jeff. That's good. Would you mind illustrating again one more time?
Yeah, you're aiming right for it.
Okay. Stretch my jaws around.
Oh.
All right.
Can we get a tighter zoom on that, Jeff? Yeah, so the underside of this rock has this kind of crumbly texture to it. Very nice. If you could rotate, Jess. Yeah, thanks. I have a feeling this is going to be similar to some of the ones we've collected previously, but that's fine.
Okay.
It's good to get another representative sample from the site.
So should I put it back?
No, you can go ahead and keep it.
Oh, Roger.
We'll put it in the starboard, not the starboard, the forward bio box.
Roger that. And I'm assuming there's nothing in those.
Correct. Okay. Well, at least not that I'm aware of.
Nothing. Yeah.
Roger. Thanks, Jake.
Yep. And before we pick up, if we could just tilt the camera up a little bit to look at some of the other rocks in this area before we move away.
Sure thing.
What's the sample number there, Steve? I forgot. 90?
90.
So I just want to see what's kind of up in this direction.
Do you need an eDNA at this site?
That would be a good idea, yes.
Rog. Is that a good zoom for you there, or a good view?
Yes, thank you. I just want to get a better idea of kind of the in situ look of the rocks here to convince myself that that sample was a good representative. And I think I have. Okay, thank you.
All right, full way, please.
Let's go ahead and get an eDNA, please.
And what's the idea behind getting water along with the rock sample?
So one of the goals of one of the projects of this expedition is to understand the microscopic life living on these rocks. And we want to collect water samples from these same areas so that we can see what the continuity is or the differences are between the microbes that might be living in the water and doing their own thing and the microbes living on the rocks and doing their thing. And then some of this water will also be used for the broader eDNA study to see if there's any evidence of larger life forms shedding cellular material into this water. It's not a really, really dense community, but we can see in Argus Cam that there's probably some corals and sponges up ahead. So.
Okay.
Popped.
Right.
Fired.
5 is fired.
5 is fired. Is there any interest in getting a picture of this? Is it an anemone in the sediment?
Sure. Yeah, we can quick zoom if we have time.
Yeah, let's do a quick zoom on that. And patiently waiting for his photograph.
Would you like me to call that move in now?
Yeah, we can go ahead and
sure.
call in the ship's move too, Rennie.
Go ahead and push on in there, please.
Bridge nav. (100 meters, 130).
Yeah, this doesn't look like a true tube anemone to me. This looks like an anemone that just is living on or just slightly under the sediment surface. It doesn't have the inner band of tentacles around the mouth that's indicative of the cereanthids. So, anemone.
But pretty nonetheless.
Yeah, exactly.
Thank you.
All sure thing. Full wide.
Yeah, so there's a couple bamboo corals around us, so maybe the eDNA sample will have some, some things in it.
We have like about a minute or two. Do you want to push it on this one here?
No, I don't think we need to. We can go ahead and move. It was more just a frame of reference for the sample for Meredith.
Roger. Someone is wondering if the water sample could be contaminated by the collection of the rock.
That was a very clean rock collection in terms of not kicking up a lot of disturbance. And we can see in the camera that there's already some particulate matter in the water. And so the rock sample probably would not contribute enough to make a difference. But it is something we always want to be mindful of. So for instance, when we're sitting in an area, we also have to think about the disturbance that the vehicle is causing. So if it looked like there was a lot of sediment in the water, I would say, don't collect the sample here. Being sediment that was generated by the vehicle thrusters. On a previous dive, not on this seamount, but on one to the east of here, as we were transiting the vehicle over the seafloor, we saw a fossilized rostrum of a beaked whale.
That was so cool.
Which is a really exciting find. And so pointing that out to the audience, always keep your eyes peeled as you're moving across the seafloor, because you never know what you're going to see. And may look like the same thing over and over again, but every now and then there's a surprise. So we can see in the view from Argus, again, that there's kind of a slope here that's relatively sediment covered. So whether that's an indication of difference in current in this spot that allows the sediment to build up, so maybe the current's not as strong here, or it's angled in a different direction. It's important for us to think about those things when we're trying to understand the diversity and density of animals. There's all kinds of features, variables that can go into that. It's not just water depth, but it could also be a reflection of rock type, slope, orientation of that slope, current, food delivery, all of those things will impact the density of the animals.
Jeff, you want to do a partial on this? Oh, never mind.
Hi.
The dancer is up the building.
I mean, it looks fun, though, doesn't it? Just drift down to the seafloor.
Graceful landing.
Very, very graceful landing. Is that a polyp medusa on the left there? On that primnoidus?
On the coral.
Oh, yeah.
I suspect it's an anemone.
Anemone.
Anemone. Yeah.
Anemone.
Not the right kind of coral for it?
Well, so those coral-eating polyps I mentioned, we tend to find them on chrysogorgids, and I don't know, particularly Iridogorgia seems to be where we've collected them most. It doesn't mean that's the only coral they choose to prey upon, but
brittle star, it looks like.
The brittle star, yeah. Yeah, but that's a good question. It's hard to say definitively that medusa only preys upon Iridogorgia, but that's where we primarily find it.
Mm-hmm. Someone's curious if of the rocks that you've sampled thus far and looked at, have there been a lot of anaerobic bacteria on the underside specifically?
So that's not something that's easy for us to determine on the ship. It'll be easier to figure that out when we're back in the laboratory. Undoubtedly, there's going to be some little pockets of anoxic conditions within the rock
white crab
structure.
there?
White crab on the left, lower left.
Sorry?
Just out of the bottom of the frame, left side, there's a white
crabs. We
There
can do like a snap zoom.
Oh yeah, there we go.
Yeah, let's do it.
And I'll tell you if it's worth.
The other side, don't we? Let's try here. Go ahead, Jeff. You want the other side there?
Can you, yeah, can you, no, it's another
squat
lobster, yeah. Yeah, thanks though.
Very spiky, very cool.
Yeah, it actually looks like Munidopsis bairdi. It has this very long kind of rostrum that it curves up, kind of like a rhinoceros horn.
Oh, anyway, back to what I was saying. Yeah, so there might be some anaerobic bacteria in the rocks, but it's not possible for me to determine that out here because there's not a telltale signature for what that would look like. But we've preserved the samples for investigating in the lab, and we'll see if there's any anaerobes in there.
Great, thank you.
Thanks for the question.
Could see some sort of feature coming up on sonar, just a little rise, and then kind of seems like the coral that we're seeing in Argus can have taken advantage of this advantage.
Yeah, so again, kind of on a sheet flow, but with a different orientation. Must be stable enough food delivery to allow a slightly higher density.
This is another type of bamboo coral in front of us. It's a keratoisid.
It's got that interesting gap in the middle. What?
Yeah.
We think that it was parasitized and.
Yeah, some kind of predation maybe. Yeah. You can see the bare denuded skeleton in the middle. It's falling apart. But this is probably in the J clade for bamboo corals.
Oh, a partial zoom there, Jeff.
There we go. So it looks like branching at the intern nodes or just above the node in each case. But yeah, this is very reminiscent of J clade.
All right, pull away, please.
Thank you. Yep.
All right, yeah, let's keep moving. So as a reminder to our audience at home, one of the motivations for today's dive was to see when we get into an area where these sponge get into really high density, which is something that we saw on the previous dive, on this same feature of Don Quixote seamount, but at a slightly higher depth, water depth, slightly shallower water depth. So the goal of today's dive is to start at a lower depth and work our way up to where the previous dive started and find out where that point is. So far we're not seeing a super high density of sponges, seeing a fair density of corals, but I envision as we get up into some shallower depths, we'll start to see them. So Rennie, what kind of ship's move do we have currently planned here? Is that going towards the like 746, 742?
Yep, 746 should be the where Argus settles.
Okay. Can you zoom out just a little bit?
Sure thing.
Yeah, great. So I think once we get to that 742 mark, let's go ahead and start making that northeast move.
Something like that.
Exactly, yeah.
Can do.
That'll be interesting to see if that sponge density is high on those little pinnacles up there compared to what we saw on the flank.
As I was asking about advice on what subcategory of biology degree to get in, exciting. You can definitely check out all of our biologist backgrounds on their bio pages. I would encourage you to click through there and see what subcategories they studied.
Yeah, and I guess it's a question of what kind of biology you're interested in. So if you're interested in kind of invertebrate biology, if you're interested in microbiology, if you're interested in, you know, fish biology, there's many different tracks that one could take.
She says she wants to, or they want to do what you do.
Oh, okay. Yeah, so then you want to go into programs that offer either like environmental microbiology. A lot of programs have general microbiology, but those are more like clinical microbiology, you know, about like human microbiology. And so the kind of stuff that I study, I actually never took kind of a standard microbiology class. I always came into it from a geochemistry and environmental sciences background because my interest was in the role that microbes play in the chemical cycles and less so about like the inner workings of the microbes. So it kind of just depends on what you're really interested in. And then over time, I've gotten really interested in genetics and genomic analyses, but I am not an expert in those things. And so I rely heavily on my team who does have bioinformatic experience to look at those genomes. And so that we can approach the same question, but from different angles, me being a chemist and they being a bioinformatics scientist.
Thank you.
Yeah, and thanks for the question because it allowed me to use my hands a lot, which is helpful.
I know, I'm sorry, not everyone could.
In the room. Okay, so left behind some of that more exciting basalt and sheet flows to kind of a more flat area. Doesn't seem to be as highly colonized, a little bit more sediment here. But our trajectory has us getting a little bit shallower soon, so we'll see how that changes.
There's a white crab.
Yeah, white crab on the left.
Oh yeah.
Oh yeah.
You guys are on crab patrol. I like this.
It's got a very squat lobster silhouette though from this distance.
Yeah,
so.
it does. Kalamaehu, can you help us with, or help me? What is the name for crab in Olelo, Hawaii?
All right, go ahead, Jeff.
So, I'm not sure what the applications
A
might be
little
for
wide
these
there.
specific species, but
Yeah.
papaya is going to be the general term for like crab and stuff like that, and then
Yeah.
ula is going to be lobster.
You want a different
Ula?
view there, Steve, or can you
Okay.
identify it from this?
Yeah,
Thank
can
you.
you push
You're
in
welcome.
as good as you can get with this orientation? I think this is going to
It's
be
got
our
that
squat
same
lobster
rhinoceros horn.
again. Yeah, yeah. So, yep, no, not the crab we're looking for.
Roger.
This is not the crab you were looking for.
Yes.
We're all thinking that, aren't we? This is
Yep,
not
every time,
Yeah,
every time it's said.
time. Steve, what are you qualifying as high sponge density?
Oh, it's very subjective. So, there was a paper, I think, in like an internal NOAA report I saw recently about what might constitute a high density or high diversity community. But I don't recall the exact metrics off the top of my head. I think it's just you'll know it when you see it type of thing.
Okay.
You know, there's nothing in the literature that, that says like in a high density community has to be so many colonies per square meter.
Right.
There are terms like coral garden that are well defined as, being so many colonies per square meter. But, it's not necessarily, this is not necessarily a hard definition, high density or high diversity.
What is the metric for coral garden?
3.85 colonies per square meter.
That is very specific.
Yeah. Yes.
Okay.
It was in a paper in 2006 that was from the
what is that?
Gulf of Alaska.
Is that a sea cucumber?
Oh, yeah.
Over here to the left? Whoa, I don't know what I just changed my mark there.
You should put it on one of the, there's a lot of cartoons in there, in the telestrator.
Yeah, there's some emojis in there in the settings.
Oh.
Oh, don't tell me that.
No,
Get
yeah,
dangerous. There's like little lobsters and crabs and stuff, fishes.
Interesting.
Somebody got rid of those. I added them to the
What?
drop down list last year, but somebody.
All right,
got
go
rid of
ahead,
them.
Jeff, and push on in on the health area.
It's probably just a setting.
That's good.
Went to the default.
Sea cucumber
And shrimp.
and shrimp. Yep.
Kind of
This
looks
one
like a
looks like-
tick from this angle.
Like a what?
Like a tick.
Okay.
Sorry, Beth.
I'm not offended, don't worry. I mean, I could be offended on the sea cucumber's behalf, but
Yeah.
okay, thank you.
Swooping forward.
We do have a standing request from some scientists at UH for sea cucumbers, but I don't think that's one of the ones they were looking for.
Okay.
Ooh.
There's 4 particular species, and a couple of them are sea pigs. That one wouldn't, I don't think, fit the definition of a sea pig.
No, it didn't look like that.
Yeah.
Kalame, there are concerns that you got cut off. Would it be possible for you to repeat what you were just saying? Rennie, you are on it. I think maybe the
What about
the
the
crab?
crab and
White
lobster
crab
names?
on the right.
Yes. So just a general term for crabs is going to be like papa'i and stuff like that, but I think that more so pertains to like shoreline species. And then there's another general term for lobsters, which are going to be ula. An interesting point to make that a subcommittee within the cultural working group is the nomenclature hui, which is comprised of representatives with longstanding and various sort of like stakes within the monument. And so part of their responsibilities are to discuss possible Hawaiian names for some of these species and stuff like that. They recently just did a presentation on some of the bird and other fish species that just came out too. So I would not be surprised if, or I would hopefully encourage more conversations around some of these species as well.
Right, the deep sea ones.
All right, full wide there, please.
Ula. Not papa'ea.
Nope. Yep. So yeah, papa'i is going to be crab, ula, lobster. And then it's interesting because now I'm looking at like the various words for sea cucumber and stuff like that. And so trying to be able to like cross-reference some of these things as like, maybe like these as like preliminary ideas for like nomenclature, which will of course need to be decided by the working group and nomenclature hui. But it's been very interesting. I think we've been having actually big thank you to Steve because he's been giving me a lot of, well, I'm learning a lot from everybody, but just talks about like the deep sea corals and stuff, and then like being able to, put that within a framework of like Hawaiian language and stuff is very interesting. And so,
all right, Jeff, go ahead and push in on this guy too.
Like I've been saying, a lot more questions than
Whoa.
answers.
Whoa,
so
cool.
Backwards, squatty.
That's also a squat lobster, right?
Yeah, both.
They heard us talking about them.
That's it. Yeah, same squat lobster. So
Roger that. Full wide.
If we do find the one that
we're gonna scream.
I'm impressed.
well, it's just like it's like a decoy.
Oh,
right?
yeah, I'll say something. Yeah, I'm impressed at the number of squat lobsters, though. I didn't expect them to be that
There's
many here.
two more over here.
Yeah, there's another set over
on
there.
that sponge.
Because I don't think I've seen this many in any other dive we've done.
I wonder if it's just because we are looking
do
for
a
them.
quick partial
And
there.
the other dive, it was so dense with everything else that we just overlooked them, especially
And on
because
the
they're so
rock
white.
too.
Yeah, the rock one
Yeah.
looks different.
The lock one does look different.
And that's
Maybe it's
Still
smaller,
squatty.
smaller version.
Yeah.
And the one on the sponge up there?
The one on the sponge. Let's see. That's the rhino-y looking one, hey?
Yeah. Rhinoid, I like that.
Yeah, I agree.
All right.
I'll have to go back and find a photo of this for the future dives because I want to be able to convey the characteristics we're looking for. But it's not easy.
Is there a photo that we can?
Yeah, I'll try and pull one. Yeah, I'll have to go back.
Let me know when you have it up. I'll bring it up for Jess.
There's so many now.
Because now there's just a whole ton of white.
Yeah.
They're
White crystals.
everywhere.
Yeah, we're gonna need that photo.
Trying to find that, that
Go
one
and
photo.
partial zoom there.
Yeah, among
that one's up.
thousands may take some time, so I'll have to get back to you.
That looks.
Yep, more
Yeah.
Unitopsis.
We, I know some of the science communication
Go
fellows
wide.
have spent some time looking through the stills, so maybe they remember what dive.
This is a cool shot, though.
Yeah, now, so it looks like we're transitioning into a slightly different conglomeration, density, and rock type. Well, density's definitely picked up. So we're at a depth now of 2,060 meters. Seeing some more sponges. Larger diversity of sponges, diversity of coral. Go ahead, Kalamea.
Just a quick note on the ula, on the lobster thing. I actually mispronounced it, so there's no okina there. Ula is going to be the color reddish, and then ula itself is going to be the lobster. So sorry, Gengi. I'm going to check myself real fast.
As a very unnuanced person trying to understand Aleho Hawaii. I didn't quite get the difference between ula and ula.
So ula itself is going to have the okino or the glottal stop.
Yeah.
And
So
maybe I'm not announcing it. Yeah, it's going to be that glottal stop where like it looks like a backwards apostrophe.
Okay.
And then ULA.
Okay.
Whereas the term hula for lobster is going to doesn't have that. I might not be enunciating that well.
Okay.
But
okay.
Jeff, you do partial in this
Thanks,
card?
that helps me visualize the difference. Thank you.
That's great there. Nice bunch.
All right. Did we lose the lobsters? Are we just not seeing them anymore because now we're looking at sponges and corals?
Well, all these Chrysogorgia colonies too have squat lobsters in them, right at 6 o'clock here.
Yeah.
Yeah.
So if you went and looked at all those Chrysogorgia, you'd find multiple squat lobsters in there.
But they're often the
like
red or pinker ones, right?
Yeah, yeah, they're usually chirostylids, so a different family.
Yeah.
Yeah.
So not the white ones. We want
Not
the white crab.
Yeah.
Where's the white
those
crab?
are Galathioids. In the Munidopsidae, I think is this family.
You said they live on dead sponges usually there, Steve?
Yeah, I think I saw it on a sponge last time.
So the ship has completed its move. Argus has another few meters to go.
Okay.
Gonna call in 12075. Start to head up that slope.
Roger that, 075.
Yes, that sounds great.
You can do partial. I can't tell if that's just... Oh. Okay, full wide.
Barnacles. Yeah?
Do you want me to come to 075, Jess?
We can wait a minute here while
Okay.
Argus finishes that move and then head on over. Thanks, Jake.
Yep. Gonna do a gauge check as well.
Sure thing. Sponge.
Renato, which screen do you use most for navigating?
I find that I often sweep all
Can you
of
do a
them.
partial there, Jeff?
Yeah, it's a bit.
Great.
We have the overall positioning in relation to the features we're looking at, kind of a top-down view. That would be like what you think is a more traditional map. But those scanning sonars really help as far as what's actually happening. You know,
Okay,
that's going to be
forward
much
there,
more locally accurate to what we're seeing down there. So you don't want to get lost in the weeds on a map and then slam into a wall.
Right. Rennie, do you have the ability to see Fledermaus in here?
Yeah, I can pull up one of those computers.
There was a really nice view shown in the lounge earlier. It's kind of a side view in Fledermaus that actually showed
There we go.
the dives.
Yeah. So this is the dive we're on. This is MB Proc.
All right, stand by.
Should be MB Proc 1, 2.
1 second as we bring that up into channel 3 so that you can see what we're seeing.
Yeah, we can do cool stuff. We'll start from the roughly north.
All right, Jake, go ahead and turn your heading there.
Roger that.
One second.
Looks like
There's so many,
Jeff
so
have
many
that
choices.
up. Yeah.
Not
Not
quite
yet.
yet.
Not quite yet. Okay, sorry. Just saw some blue over there.
I've only got 4 different MB procs to pick from, so
Oh yeah, you don't. Yeah,
it's probably
it's the MB Proc
that
regular.
or that. There we go.
There we
Okay.
Last one that I'd pick.
Yeah.
Thanks, Jeff.
So here we go. Here's a lot of the seamounts that we have in this general expedition, starting from the southernmost and going all the way up. This is on channel 3 on the quad, by the way. And
Yes.
you can see we actually dove all the way down here. This was our first dive, but I'll focus as we go up. Had a couple dives on this seamount.
Yeah, hhhmm.
On different sides of the slope. One on this little mound.
Jake, you want to come up a little
Another one
bit?
on the north side of Don Quixote. And now, as I zoom in here, we're on this section, this white line that you see. That is actually our dive track from the prior dive. And these two pink diamonds are the start on the west and the end on the east of our current dive. So we don't have the active dive track in there. That's what we're using HiPAC for. But if you want to see, as I zoom in here, I'll give a little bit of a perspective view. There you can see that's the descent. The descent of our last dive.
Yep.
And then right down to the seafloor, and then we tracked along that ridge, zigzagged up, and then ascended. So
yeah, and in that view, that white line, we saw pretty high density of coral and sponge that whole time. So part of our goal of going between those two pinkish purple diamonds is to see when does that high density start. Thank you so much for showing that.
Sure. If you're wondering why there's a bunch of different colors in there, we are mapping on the fly, so we haven't quite meshed all those grids together, but we like to see the coverage, so the color bars don't exactly match, somewhat purposefully, so we know what we've just covered and what the prior expeditions have covered. But this is mainly what I do all day, is just kind of zoom in on this kind of stuff and do that.
And it's a nice view, too, to see that unnamed seamount to the east of Don Quixote where we did a dive, which is, you
this
can
one
see,
here.
Yeah, exactly. Didn't have enough volcanic activity to create a tall enough volcano to then breach the surface of the water to form a flat top guyot, which is what we're diving on today.
Flat top over here.
So
those ascents and descents are pretty interesting. You can see the scale of our bottom track. You know, we try to get down and up as soon as we can, but once we're down there, we really take our time. So you can see that as far as distance covered, it's roughly equal to one descent. But
yeah. And most of our dives have been similar lengths of time too, so.
Jess, there's your ascent where we do the zigzag.
That is pretty nifty.
That's cool. That'd be a cool one to 3D print. That whole seamount.
The sense.
Yeah.
Yeah.
That's very cool. Rennie, folks are wondering why we can't get a live view with that.
You can. That can be fed into this software. It's just a little bit finicky, and sometimes we find that it's visually stunning and cool, but it's not always the most useful.
Yeah.
So it's kind of, sometimes we'll do it, but it requires some loops and it fails sometimes. So.
Thanks a lot for that, Rennie.
Sure.
So coming back to the live views of the seafloor, you can see here that we're back in a little manganese nodule area with some other small rocks that seem like they've maybe rolled downhill at some point a long, long time ago. And every now and then we see an animal attached to one of these larger rocks. Sometimes it looks like they're falling over. Not as many lobsters or crabs. Lindsay, can we tell where people are tuning in from tonight?
Yeah, we have folks from all over the world. Got folks tuning in from, really all over the world. A big group from South Africa, Australia, New Zealand, quite a few all around Europe, few in East Asia. China's represented here. And then North America, of course, well represented, Canada, USA, Barbados.
And then Hawaii actually has quite a few tuning in tonight. Please, we would love, since we're in your neighborhood, let us know where you're tuning in from in Hawaii. How did you hear about us? Send a special note for Kalamehu.
Yeah, and we must have some late night warriors on the mainland of the U.S. It's got to be in the middle of the night to be tuning in, but thanks for joining us.
Or early risers getting ready for Black Friday shopping.
Oh, good grief. Don't need to be reminded of that out here.
Well, if they're waiting in line and watching Nautilus live, that is pretty cool.
Yeah,
Yeah,
that's true.
dedication.
Someone in England is drinking their morning tea and watching.
Okay.
Thank you so much for the color.
Yes. What a nice calm way to start your morning. Jess, while we're floating by, can we get a partial zoom of this black rock that is suspiciously uncovered?
Yeah, sure thing. All right, go ahead, Jeff.
Yeah, interesting. We've seen this a couple times on this cruise where we'll have these rocks that just have a really shiny texture on the top surface.
I want to know what
Is
the
that a
thing
coral
off the left
patch
side
on the
is.
back side?
Yeah,
Yeah.
there's just like a
Is that some
bubblegum.
zoanthids on
Looks odd.
zoanthids on an old stock?
Looks
Yeah.
like it, yeah.
Could we, Jeff, can you get in any closer on this?
On the rock, or do you want the stock?
Yeah, on the animal.
I think
Prepare to be slightly seasick. Just turn my heading
I
a
think
little.
Rennie wins the game there.
Yes.
What did Rennie
Looks
say?
like zoanthids.
Bingo.
Zoanthids or anemones. They could be anemones also. On an old coral stalk, like a branch of a coral. That
There
is,
be any interest
All right,
in
Jeff,
that?
go ahead and push in a
it's
bit more.
So small.
well, I mean, if you want the rock, yeah.
I'm indifferent.
I mean, it's an
Okay.
interesting observation, but I'm not going to collect
Yeah.
that just for that.
Okay. Thanks, Jess
Pretty
and
neat.
Jeff. We can keep moving.
And of course, a mushroom coral off to the other side.
Steve said I was right about something, so I'm going to be riding that high for the rest of the year.
Credit where credit's due. I'm going to put that in the event log. Rennie was right.
Yes.
Did you want the hero shot?
Preserved in time forever.
There we go. There's the hero shot.
Yeah, so I think the kind of base it was something like this Paragorgia colony coming up. I think that's what the base was, and then it was just covered with zoanthids.
Okay. Yeah.
I concur, Steve.
Indeed.
Again, we're seeing some of these remnant stalks.
Go ahead and push on in there a
things
bit
that
on this guy.
are no longer with us.
Very pretty.
That's a type of bubblegum coral.
Steve is a coral doctor, and I just play one on the radio. Or whatever it is this is that we do.
All right, full wide, please.
You can't see it right now, Randy, but Steve is actually signing both your bingo card and your official degree, so congratulations.
And again, some suspiciously shiny rocks.
Interesting pattern in the
Sediment?
yeah.
Yeah.
I don't know if those are slides or what.
Yeah, it almost looks like ripples.
Yeah. And like the finer, the rougher stuff is right at the edge of it. And
yeah. I mean, yeah. Low, like sediment sorting.
yeah. Yeah, you would expect current to do that, I guess.
Almost looks like scoured in a way, but
Yeah, there's not
quite.
much of a slope here that would cause that, I wouldn't think.
Yeah.
It was the critical angle for internal wave generation.
The view in the sonar for Argus is very interesting.
Yeah, I'm curious how much of that is sometimes we have some interference that causes some patterns that look like that, but it also could be real. Oh, now this is interesting.
Yeah, that texture there.
Yeah.
Yeah, it's like just a river.
Oh, that's cool.
Wow.
A very distinctive
It's
line.
almost like a wind-blown beach, like
Yeah.
That's a wicked shot in Argus there.
like
Yeah,
like a dune.
that's a good context shot.
Yeah, really good. Yeah, you can see that.
Super cool. It looks like that'd be like a sheer wall, but it's actually just sediment.
Yeah.
Cool.
Well, I wonder if the substrate is actually if there's a flow that's causing the darker things that we're seeing in the sediment is what's backfilling it.
Yeah.
Or if it's something that, like we were discussing.
Jake, you want to frame up that shot in Argus and then we can maybe get a zoom in there? It's cool.
The rare Argus. Yeah.
Jeff, you can zoom
Yeah.
on Argus.
That's great.
Nice. That's cool. Nice.
I didn't know that we could do that. I'm going to ask for that more often.
Yeah, sometimes when we're sampling, when we get a nice Argus zoom and you can see the arm doing its thing.
Cool. Nice. Thanks, guys.
Nice. Good shot.
Yeah, it's almost like terraced.
Yeah.
Mm-hmm.
Huh.
Oh, there's some more interesting sediment textures.
Can I come up a little bit there, Jake?
Yep. Coming up.
Maybe those ripples are indicative of the flow hypothesis.
I have found the crab, but I have to double check.
It's quite a colony here.
Yeah.
Yeah, you can see a nice, I guess that base is probably about 15 centimeters.
This is radiolysid, again, J-clade.
Much healthier looking than the last one we saw.
Yeah.
Had a big gap in the middle.
Can I do a slight partial in on this guy?
These are usually the bamboo corals that are typically overgrown by the zoanthids, too.
Very cool.
It's good to see
Nice
that
scene.
not all of them have zoanthids on them.
Yeah.
Looks like there's a little shrimp swimming towards us.
Oh, you can do it.
Full wide, please.
Thank you.
Oh, and a fish
And a
off in the
fish.
distance.
Oh, yeah.